Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. As you guys know, these videos are intended to provide a basic and foundational approach to some of the fundamental principles in microbiology. By no means is it a comprehensive review of all the information that could be covered. But if we provide a solid foundation, then later on as we add more and more detail, those details will fall in place. Now, if you're taking this class with a different instructor, then you need to make sure you cover the material that instructor wants you to know in the manner and the fashion that instructor wants you to understand it and learn it. So today's video is going to be over DNA structure, heredity and genetics, and protein synthesis. How do we use DNA to synthesize proteins? Rather complex topics, far more detail than we can get into. But this should provide a solid fundamental understanding of the basics behind it. Now, when we talk about genetics, we need to define a few terms. As always, we need to have our definitions in place so that as we discuss things, we know what we're talking about. So, genetics itself is often defined as the study of heredity. Okay? And that's really a very broad definition, but heredity is talking about those inherited characteristics that one generation receives from the previous generation. No matter what type of cells that we're talking about, because cells come from previous cells, the new daughter cell that is produced has some of the same, if not all the same genetic information as the parent cell. And depending on how cells divide and reproduce, it can be altered somewhat. Sometimes it's very pure in the sense that we transfer identical genetic information, like in bacteria. Usually, if there's no mutations, the bacteria will produce identical clones of each other. But in humans and most animals, and because we have sexual reproduction, usually you have genetic information, hereditary information being passed from two parents into one offspring. That offspring exhibits a mixture of the genetic information or the hereditary information. But essentially what I like to think of this is it's the study of genes and how they function. It's far more than that. Okay? There's no real easy way to put a simple definition on genetics. But we can start with that. Now the genome, when we talk about the genome of an organism, we're talking about all the genetic information in that organism. Or sometimes we say in that cell, because some organisms are single cell. Now, for most eukaryotic cells, or really essentially all eukaryotic cells, the genetic information, the DNA, is contained inside a membrane-bound organelle called the nucleus, where in prokaryotes, archaea, and in bacteria, there is no nuclear envelope or nuclear membrane. There's simply a single circular chromosome. In eukaryotes, the chromosomes tend to be long and linear and have multiple genes on them, and there's many, many chromosomes. Where in prokaryotes, there's a single chromosome and it's circular. Now, as we're going to learn a little bit later, prokaryotes can also carry some extra chromosomal DNA or some DNA outside the chromosome. For example, if we look in bacteria, they usually have a single circular chromosome. And the area where the chromosome is found is called the nucleoid region. You'll hear us talk about the nucleoid region or the nucleoid. And that's an area sort of where the DNA is found. It's not a nucleus. It does not have a membrane around it. It's what we call the nucleoid region. And let me grab my eraser. Now, when we talk about this, or when we talk about bacteria, one of the things that we know is that bacteria can have another or one or more pieces of DNA outside the chromosome. Hence the term extrachromosomal. Extra always means outside of. And so outside the nucleoid region or where the chromosome is, bacteria can also sometimes have another small circular piece of DNA called a plasmid. So when we're talking about the genome, we're talking about all the chromosomes, all the genes in a particular cell or organism. And in some organisms, it includes the chromosomal DNA and the extrachromosomal DNA. And if we're talking about viruses, sometimes the genome of a virus is made up of RNA, and we'll talk about that in a little bit. Okay? So you guys need to know when we're talking about the genome, we're talking about all the genetic information inside the cell. Well, that requires us to talk about things like a gene. A gene is a series of bases, or we can say nucleotides, depending on how we want to look at it, on a chromosome that codes for the order, oops, I didn't spell order very well, order of amino acids in a protein. So when we talk about a gene, we're talking about the instructions for a single protein. A gene is literally the instructions on how to assemble a protein, one amino acid at a time. So, if I were to think about these markers that I have here, okay, I have tons of these markers. I'm going to put some more on the board over here, kind of out of your sight, just in case I need to reach for them a little bit later. Okay. I can have multiple red markers. And let's say I have a bucket of red markers, and I've got, 50,000 red markers in the red bucket. And I have 50,000 green markers in a green bucket, 50,000 blue markers in a blue bucket, and essentially I have 20 buckets. Each bucket contains only one type of marker, but there's thousands of that type of marker in that bucket. Well, those markers represent what we call amino acids. Amino acids are the building blocks of proteins. The difference between one protein and another protein is the number of amino acids and the order of the amino acids. And it is that amino acid order, the primary structure that we've talked about, that will determine how the protein folds and how the protein functions. Well, our cells can't wait to randomly assemble a bunch of amino acids. It could take literally millions of years for the amino acids to come together to form a single protein, and if we needed them to be in the right order. For example, if I told you while you're sitting here, I want you to take all of these markers, thousands of each color, only 20, because all proteins are made out of the same 20 amino acids. If I said, sit here and build me a protein, I want you to put these markers together in a long chain. Go for it. But I don't tell you how many markers I need you to use and in exactly what order. You would be sitting here randomly guessing. Does it start with green? If I don't give you any information, then you might start assembling these, and when you're done, you might ask me, is this the right protein? And I would say, nope, try again. If I don't give you any information, wrong colors, too few, too many, whatever, you would be guessing forever. There's millions of combinations and permutations with the number of amino acids. So it cannot be random. But if I gave you a piece of paper, and on that piece of paper it said, please build me a protein out of this many amino acids. It's going to be 54 amino acids long, and this is the exact order. I want you to start with red, and then there's going to be green, and then we'll put blue, and this funky color red, and then black, and then we'll do 2 greens, excuse me, and then a brown, and then we'll do another pink or whatever. Now I've built a protein. It's 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids long. Most proteins are much longer than that. But it's built in a specific order. And if I needed another ribosome, another student, for example, to build the same amino acid, the same amino acid sequence or protein, because I need thousands of them when I need them, then I could take that note and photocopy it and hand it to all the students And then everybody could be building the exact same protein, or at least half the class, and I would get a whole bunch of these in a short period of time, exactly as I need them without any guesswork. So that's kind of what a gene is. A gene is the instructions that tells us, and the instructions are written in a chemical language, and these nucleotides are bases, that tells us the exact order of amino acids for a specific protein. If I need a different protein, I need a different gene. So, because of all the different proteins inside of our cells and inside bacteria and other cells, including viruses, which are not really cells, I need lots of genes. And if I have a whole bunch of genes, I could put them all together into one long piece of information, which is what we call a chromosome. A chromosome is a long piece of DNA that contains many genes. And again, this isn't the best explanation. Some of these terms are pretty hard to define, and there's multiple definitions for them that all say essentially the same thing. But sometimes I have a whole bunch of genes that I need to link together. So it's kind of like, instead of walking around with a bunch of pieces of loose leaf paper, what if you put them all in a notebook or a textbook? And you have a textbook for all the information for micro, for anatomy and physiology, for chemistry, for history. When you need a particular piece of or particular protein made, you've got to go in your collection of books and you've got to find out which chromosome, which volume of information do I need to go to, and then which page number do I need to go to. And that sort of page of instructions in a giant book of information, the page would be the gene, the entire book would be the chromosome. Now, one of the things that we do know in eukaryotes, we usually have multiple chromosomes, and they are linear. So we'll have lots of pieces of DNA like this, and each one may have different genes on it. In prokaryotes, there's usually one circular chromosome. Okay? Also, in eukaryotes, we usually have pairs of chromosomes. We usually have 2 copies of every chromosome, and the copies are not identical. So we can have pairs of chromosomes, and so for every chromosome, I have another one that's almost identical to it, but might be slightly different. And the way I like to think about this is, let's say you were going off to college and your mom bought you a set of encyclopedias. Now, before cell phones and computers and Wikipedia and everything that you can look up on Google or whatever browser you use, we had books called encyclopedias, and there was 23 volumes of encyclopedias, 23 volumes in one set, A, B, C, D, and then some of the letters don't have a lot of stuff like X and Q, so you might put some of those together. So you might have Q, P and Q together, and X and Y and Z in one volume. So we have 23 volumes. If I wanted to do a paper on Afghanistan, I might go grab the A volume, flip to that page, and I could read all the information, and it would tell me Afghanistan's a country in the Middle East, and this is the type of government, and the population, and the gross domestic product, and the national song, or whatever. If I wanted to do a paper on Zimbabwe, I would go to the Z volume, and it would tell me essentially the same type of information about Zimbabwe. And each one of those things would be a gene, each book would be a chromosome, and I could find tons of information on each chromosome. Well, what if when you go off to college, your mom bought you the Encyclopedia Britannica, but then dad bought you the Funk and Wagnalls set of encyclopedia? 2 different publishers, same information. Well, if they had the exact same information in the exact same order, then that would be a copyright infringement. So when I want to find information on Afghanistan, I would go to the A volume, whether it's Funk and Wagnalls, Dad's version of the genes, or in the Encyclopedia Britannica, mom's version. But the information isn't always exactly the same. It's the same letters and same info, but they're a little bit scrambled. One might say this is the gross domestic product, the population, the culture, the national. The other one might start with the national flag, and it's the same information, but a little bit scrambled. So I have two versions of the same information. That way, if let's say they bought these used at a used bookstore, and one page happened to be damaged and missing, you would not be able to produce a protein if you have a damaged gene. But if you have a second copy, then you might get that copy of the protein made. So we have two shots at every one of them. Where in prokaryotes, there's usually a single circular chromosome, and there's only one version of each gene, and that's it. And when we have two versions of a gene, we call those alleles. So I'm going to write this term up here and do a few more definitions. So when we talk about an allele, we're talking about different forms of the same gene. Allele is a different version or form of a gene. For example, your mom might have brown eyes, your dad might have blue eyes. When you get the chromosome for eye color from each parent, you're going to have the blue eye and the brown eye alleles. Now there's a whole bunch of information in genetics, which we're not going to go into, as to which one gets expressed. There's dominant and recessive and codominance and incomplete penetrance and incomplete dominance and all this other stuff. So, but usually one gene gets expressed, but if that gene were bad, then the other gene might be expressed. Okay? So you have two forms of the same gene, two versions of the same gene called alleles. In prokaryotes, since there's only a single circular chromosome, you usually only have one allele. And if that gene is defective, it could be lethal for that prokaryote. Where in humans, sometimes we get lucky and can carry a disease or some mutation, but as long as we have one good copy of the gene, then we can still express that protein. Okay. And that's a real basic understanding, but I don't want to go too much more into it. I just want to get some basic definitions down. Okay. Now, we could get down a rabbit hole and spend an entire year talking about genetics alone. Now, when we talk about alleles and we talk about chromosomes, one of the things we need to talk about is this term called a genotype. The genotype is the types of, this is how I like to think of it, it's the types of genes an individual carries. Or it's the genetic makeup of an individual. It may or may not always be expressed. For example, if I have two chromosomes here, and on one of these chromosomes I have the information or series of bases that codes for brown eyes, and the other alleles for blue eyes, the genotype is this person has the gene for brown and blue eyes. That leads us to the definition called a phenotype, and the phenotype is the expressed genes, or the observable characteristics of an individual. Based on the genotype. So for example, this person would have brown eyes. That's their phenotype. You see what is expressed. If you got brown eyes and brown hair, and someone else has blue eyes and red hair, what you're seeing is the phenotype. The genotype is what genes are they carrying? What is their genetic makeup? In eukaryotes, because we often have two versions of the same gene, two different alleles, the genotype can be very different from the phenotype. Usually if you only have one set of alleles, like in archaea and in bacteria or prokaryotes, then the genotype is the phenotype. But not all genes are always expressed. Okay? And we're going to get into gene expression and gene control a little bit later. Now, I know this is a little bit confusing. There's so much information that I want to go into to try to make it make more sense and provide this understanding. But again, we'd go down a rabbit hole and we'd be here forever. Now, there is a central dogma in the world of genetics. Okay? The central dogma is the major belief, right? And when we first figured this stuff out, starting in the 1930s and 40s, when we were looking for this stuff, in the 50s, when Watson and Crick figured out the structure of DNA, and everyone was able to work out how DNA replication and protein synthesis occur, the central dogma basically said this, that DNA, this molecule, which stands for deoxyribonucleic acid. Here's your D, there's your N, there's your A. Deoxyribonucleic acid. Deoxyribonucleic is all one word. So DNA was the genetic information, the code that tells us how to make all the proteins. DNA was used to make RNA, and then from RNA we went to a protein, and then the protein gives the function. And that's the way it's believed to happen. The DNA is the instructions inside the nucleus. Ribosomes are going to build a protein based on the instructions. But in eukaryotes, ribosomes can't get inside the nucleus once they're formed, and they can't understand the language of DNA. So we have to rewrite DNA into what's called a messenger RNA so that the ribosomes could understand it. For example, let's say you hire a bunch of people that speak English, but your factory is, say, in, I don't know, Greece. And I take a whole bunch of people over there that don't speak Greek. Well, the instructions that were originally written by the engineers and everybody that design a product might be in Greek. But the employees that are going to build the protein called ribosomes can't read the Greek. So someone goes in, and then rather than having them read the instructions for every product for your factory, if I need a specific product made, a specific protein, why go through all of the volumes and all the chromosomes? Why don't I have someone else go and find the instructions for a single gene and then rewrite the information in that gene, the DNA, into RNA so that the ribosome can now understand it, like going from Greek to English, so that our English builders, our ribosomes, could understand the language and then assemble the protein. I'll give you an example at some point. Okay, so that's the central dogma. Now, we later learned that the discovery of viruses and how viruses work, we later learned that that's not always true. This information is not necessarily testable for now, at least for my students. But what we do know is that you can go from RNA as your genome. So some viruses use RNA as their genome and not DNA. Most organisms use DNA for their genetic information, and then RNA works with the DNA in order to build proteins. But some viruses use RNA as their genome, and then once the RNA is inserted, now viruses are what are called obligate intracellular parasites. That means that they are obligated, they must use the inside of another cell to reproduce. Viruses do not have cytoplasm, they do not have ribosomes, they do not have organelles. They really are nothing much more than a protein coat with some RNA or DNA inside. And that can stick to a cell, inject its RNA into the cell, or DNA, and then that somehow is going to make it into the cell and shut down the cell's productions from its chromosome of all the cellular proteins, and then force the cell to make viral proteins and copy the viral genome. And as we copy the viral genome and the viral proteins, we will assemble millions of little viruses. And then as the cell ruptures, those viruses would be released. That's how viruses replicate. They don't have the machinery to make their own proteins. They don't have ribosomes. They don't have the machinery to copy their own DNA or RNA. So they go in and they hijack the cell. They literally walk in and stop the cell from doing what it's doing and force the cellular machinery to make RNA genome and RNA protein codes, and then assemble that and release more viruses. And some viruses use RNA as their genome. When they insert, they can simply send their RNA, which would be like an mRNA, to the ribosome and get a protein. And very often these are called positive sense RNA viruses. We can just directly read their RNA and skip all of this other process and make the proteins and replicate the RNA. If the virus goes from RNA and makes another form of the RNA, uses one strand of RNA to make a new strand of RNA, which can then be turned into protein, we call this negative sense RNA viruses. And in some instances, viruses take their RNA, they have to turn that into DNA. They kind of reverse copy. Instead of going from DNA to RNA, they use an enzyme called reverse transcriptase, which we're not going to talk about right now in great detail, and then they use that to make more RNA to make protein, and we call these retroviruses. Okay? So there's different types of RNA viruses, and we might get into those in some detail later on. We may not. But the central dogma, you should know. We go from DNA to RNA to protein, and the protein gives the function. Remember, there's 4 major classes of organic compounds inside the cell. Lipids are used for cell and organelle membranes and for energy. Carbohydrates are used for energy, and sometimes it's for structural things like attaching to lipids and other things, and proteins. We have glycolipids and glycoproteins that have sugars attached to them. But we have lipids, cell membranes, and an alternate source of energy, carbohydrates, which are used for energy, proteins, which perform the functions of the cell, and then nucleic acids, which code for the proteins. It's the instructions. So, now that's kind of a basic overview of what we're going to get into in great detail. I want you to know those definitions. I want you to know the central dogma. I think you guys need to know that information. And the little bit that I've talked about prokaryotes and eukaryotes. We did talk a little bit about plasmids. Because bacteria have extra chromosomal DNA called plasmids, then we need to talk a little bit about them. So usually we have a double-stranded chromosome, circular chromosome, and we can have little smaller pieces of DNA outside that, inside the bacterium, called a plasmid. I'm going to use blue for my plasmid here. Now, one of the things we know about plasmids, now some bacteria carry plasmids, not all do, but the plasmid has what we call the extra chromosomal DNA, and very often the plasmid can contain genes that code for proteins needed for special functions. And that's a simplistic way of thinking of it, but I'm a simple guy, and so this is how I think about it. For example, we know that lots of organisms can take glucose and turn it into pyruvate in a process called glycolysis. We've talked about this when we did cellular metabolism, and out of that we get some ATP, and we get some electron carriers in the form of NADH and some other stuff, okay? Now, the enzymes that are needed to do this are inside the cytoplasm of our bacterium. Not all bacteria can, but some can. For example, E. coli, it can also break down lactose. And there are other organisms that under special conditions have genes that they don't need for their everyday function. But when we need to utilize a gene for special function, then there might be a gene on a plasmid. This may not be the best example, but I'm going to run with it. But there might be a gene on this plasmid. There might be several genes on this plasmid here that are needed for special conditions. I rarely need them, but when I need them, I need to go use them. And so what can happen is the plasmid, there are enzymes inside the bacterium called restriction enzymes. We'll get into those in detail. But restriction enzymes can recognize specific series of bases and then cut the DNA and separate it. One of them is called the GAT-C, or I call it the GAT-C. And if you look at it, these are almost always what we call a palindrome. So we have all these bases on here, A's, T's, G's, and C's, and we'll get into it. But there's a site where bacteria can come in here and cut on this particular series of bases. Palindrome simply means it reads the same way on one side as it does on the other. They are the same in reverse. Like my name is a palindrome, or the name, I don't know, my daughter's name is a palindrome. It's the same word forwards and backwards. So, very often there's an enzyme that can come in here and cut this series of bases and separate the DNA. And what it does is it leaves some overhang. Now, if that enzyme can recognize this series of bases on the chromosome, it will recognize the exact same series of bases on the plasmid. And it can literally cut this out so that when I get my cut here, I'm going to have some overlapping pieces of DNA like this. Now, if I can separate these far enough, and I have another piece of DNA, let's say from a, well, here, let me do this in a different color. So this is my chromosomal DNA. I'm going to get my cut and have some overhang here, and I'm going to separate this out to where these two pieces would have the exact matching base pair. I can cut it and put it back together. But let's say I have a long piece of bacterial DNA that has the exact same sequences. Then I could actually fuse the bacterial DNA, or the plasmid DNA, I should say, with the chromosome. And I could literally cut a little site open here and insert a gene if I need it, so that when I'm using this chromosomal DNA to make RNA and proteins, now I've got the ability to make this protein temporarily. And then when the conditions change, and if I say run out of lactose and go back to having a whole bunch of glucose, then I can cut this back out at the exact same restriction sites, remove this DNA, and put the chromosome back together. So plasmid DNA can be inserted into the chromosomal DNA or taken back out when needed under special conditions. It's kind of neat. And for some bacteria, that plasmid DNA can do things like carry genes that can break down certain antibiotics and provide what we call antibiotic resistance. There's numerous restriction sites. That's one of the ones that I remember because we used it a lot for some restriction enzymes in the lab. But for example, plasmids can carry genes for a number of things. One of them I just mentioned, called antibody resistance. And very often we refer to these as an R plasmid. If they carry the resistance gene for antibiotic resistance, not antibody. Sorry, I'm thinking of where I'm going, not where I'm at. Antibiotic resistance. So some bacteria can resist antibiotics that would normally kill other bacteria. And that's what happens when we get an antibiotic resistant strain, is that they can carry an R plasmid, and they can insert that in when they encounter the antibiotic, and they literally break it down so it doesn't kill them. Okay. There's another one. They can carry genes for what we call for the pili, and we call this the F plasmid. So some bacteria have the genes, they have a genes for what we call fertility. And so the pili provides for fertility. Bacteria don't have sex. They do what we call conjugation. And what happens is, if I have a bacterium that has its normal chromosome and carries an F plasmid, then when it incorporates that gene, it can make a pili. And if you recall, the pili will allow what we call an F positive bacteria, the one that carries the F plasmid, to fuse with a bacterium that does not have that plasmid, and we call that an F negative bacterium. And what that allows is the pili will attach one bacterium, the F positive, to the F negative, and then we can copy the plasmid and transmit that plasmid to the F negative and convert it to F positive. And then when these two separate out later, now this one has the ability to make the pili and can transfer the plasmid to the next one. So when we talk about the fertility plasmid or the F plasmid, they code for genes that form a pili and allow the transfer of plasmid from one bacterium to another. That's the closest that bacteria get to having sex. So we call that conjugation. You can think of like conjugal visits in a prison. So some bacteria carry plasmids for special metabolic enzymes. So there are certain conditions where a bacterium might need to make a particular gene product to metabolize some particular substrate. Like if a bacterium doesn't have glucose present but does have lactose, if it has a plasmid that has the enzyme to break down lactose, then it can insert it, break down the lactose, and survive even in the absence of glucose. Okay? And then finally, some bacteria carry plasmids for toxins. We call those endotoxins sometimes. And this leads to pathogenesis. So some bacteria can insert a gene in here that allows them to make proteins that can be released and that are very, very toxic to the surrounding environment. Okay? So, when we look at the genetic makeup of some organisms, particularly prokaryotes, we need to make note that they have the ability to carry extra chromosomal DNA in the form of a plasmid. The plasmid can carry genes for a number of special conditions. For antibiotic resistance, called an R plasmid, you should know that. For a conjugation of the fertility genes, called the F plasmid, allows them to form pili and transfer DNA to what we call an F negative bacteria and transform it. We have special metabolic enzymes that can be produced only under special conditions, or certain toxins can be produced from time to time. So, all right, now, before we get into the structure of DNA and DNA replication and all of that, I hope you guys have somewhat of a basic understanding of some of this information. I'm going to stop this video there. I want you to know all the definitions. I hope you guys understand a little bit about extrachromosomal DNA, and certainly that when we talk about prokaryotes, the genotype or the genome includes not only the chromosomal DNA, but the extrachromosomal DNA. Eukaryotic cells do not have plasmids. They do not have extrachromosomal DNA. They only have the nuclear DNA that codes for proteins. Know the central dogma as well. All right, listen. I hope you learned something. I hope you had as much fun as I did. The next video we'll be talking about DNA and structure and RNA structure and in a little bit more detail. And once we understand that, we'll be able to understand how we can use DNA and RNA to build proteins. Thanks for watching. I hope you had as much fun as I did. I hope you learned something, and I hope to see you on the flip side in the next video. Hit like.